Okay, apologies everyone, I think we've fixed the problem now. You can see slides, right? Yep. I'll make sure I keep checking behind me. So I won't start from the beginning again, I'll start the overview slide again. So I'm going to sort of cover why we'd want to use model organism phenotype data to understand human gene function. We'll talk about the largest project collecting mammalian genotype to phenotype associations, the IMPC and the disease models emerging from that. And a lot of the data and the ontologies and the software that allows us to do this work comes from the Monarch Initiative. So I'll briefly describe that and then finish up with this, how we use it in sort of disease diagnosis and discovery programs, bringing in the clinical phenotypes. So starting off with why we want to use model organism phenotype data. So of course, the whole reason that some species are considered model organisms is when you look at their genes function and what happens to the phenotypes when you disrupt these genes, it very closely mimics what you see in the human disease. So there's a nice example of this from one of the first Monarch Initiative papers, where you see in these PAX6 mutations in human, mouse, fish, and the fruit fly Drosophila, you see sort of abnormalities in all of the mutant forms here at the bottom. And you'll sort of notice that the abnormalities are slightly different in the different species, which is something I'll come back to later in the talk. But you might sort of still say, well, I want to understand human gene function and disease, so why not just use the human data? And the big argument here is that we only really know about sort of single gene disruptions and the phenotypic consequences from Mendelian disease gene associations as captured by OMIM and OrphaNet. So if you look at the numbers for this, there's about 18% of the protein coding genes are involved in the disease gene association in their Mendelian disease. So there's 3,367. I think this number's gone up to something like 4,000 now, but the picture remains the same. If you look in the mouse, so we have all sorts of gene editing technologies, as I'm sure you all know about. You've all probably read about CRISPR and Cas9, so it's revolutionized the field. We can very quickly disrupt a gene in the model organism and study the phenotypic consequence in vivo. So hence, there's a lot more genes covered in the model organism. So for mouse, we have, well, to date is almost 12,000 now. So over 60% of the protein coding genes, we have a mouse model with some phenotype data that can shed light on the functional consequence. So traditionally, a lot of this data came from people publishing papers and the MGI team at the Jackson Laboratory curating. More recently, a large proportion of this data is now coming from the International Mouse Phenotyping Consortium. So in practical terms, what this means, though, in the 100,000 Genomes Project, if we find a de novo mutation affecting one of these genes in this fraction, we can look at the mouse data to really assess, is this a likely candidate? Do the phenotypes look similar to the patient's phenotypes? And as I say, so the International Mouse Phenotyping Consortium, or IMPC for short, is really sort of producing the majority of this mouse genotype to phenotype data nowadays. So IMPC consists of all the sort of major mouse production and phenotyping centers worldwide. So there's sort of 14, I think, to date, and it's kind of expanding all the time. We're always looking for new partners, so we're going to bring in a South African partner soon. And then there's the informatics teams at MRC Harwell, the European Bioinformatics Institute, and my team at Queen Mary University of London that sort of collect together all the data and analyze it. And I should say this is obviously like a major sort of concern. It's involved hundreds of millions of dollars of NIH funding and funding from other sources. It's not cheap, but people see the power of it. And when we started the IMPC, the context of it was, I mean, this is surprising to some people, I think, but we still don't know the function of the majority of genes in the mouse and indeed the human. And even when we sort of do new functional studies on a new gene, often the results are surprising, like genes are way more pleiotropic than we may have predicted. So you're seeing phenotypes and systems you wouldn't have predicted. When we started the project, there was mouse knockouts available from the literature for 30% of the genes, which kind of sounds a high number, but a lot of this data is, you can sort of describe it as patchy. So it comes from a publication where the investigators studied the phenotype they were interested in. They not looked broadly. And also this data's in PDFs, in the literature. The curation team at MGI at the Jackson Laboratory have had to manually curate all this data. So what we proposed that the IMPC was instead performing a broad-based phenotyping pipeline on all genes to produce a resource that would be hopefully be useful for all researchers, but particularly the human genetics community. And obviously the goal is to build this first comprehensive catalogue of a mammalian genome. So we're going to generate a mouse mutant for every gene in the mouse genome on a single inbred background, black 6N, and comprehensively phenotype these mutants to determine the developmental, physiological, and biochemical parameters. We collect together all the raw data and run a statistical pipeline to identify statistically significant genotype-phenotype associations, and also integrate other disease sources. And the key thing is all the data is made freely available to anyone through our portal, and all the mice are available at sort of low cost through the global network of mouse repositories. So if you find some interesting data, you can order the mouse and perform further studies to look at the disease mechanism and perform therapeutic studies. So this is a high-level overview of our pipeline. So we attempt to phenotype 7 males and 7 female mice. So this is a compromise between cost and the number you need to produce a statistically significant result. And then for the adult pipeline, we phenotype between 4 and 16 weeks. And you'll see these tests in dark blue these are the tests that are performed by all centres, such as the open field test. Some tests, such as calorimetry, are performed only in certain centres. And in light blue, these are sort of new tests that are being proposed in the pipeline. And each one of these tests involves measuring several parameters. So for a particular mouse line, we're measuring over 250 individual parameters. And we attempt to phenotype the homozygous null. If it's embryonic lethal, it instead enters, so we will then phenotype the heterozygous mouse instead, and the HOMs will enter the embryonic phenotyping pipeline. So here we sort of collect gross morphology data on the embryos. We look at the LAC-Z expression in the embryos and the histopathology. But we also look at these 3 different time points, 9.5, 14.5, and 18.5, depending on when the gene is lethal. And we perform high-resolution HRM and micro-CT imaging to sort of try and really sort of capture what phenotypes are involved in the HOMs. And then looking at these same tests, but sort of broken down into disease areas. So you can see we cover a wide range of disease areas and body systems. So we have neurological behavioral tests such as open field, acoustic startle, metabolic tests. So we do many different clinical blood chemistry measurements. This sort of general tests like the gross pathology that's performed at the end of the mouse's life. Cardiovascular, pulmonary, sensory, musculoskeletal, immune, and reproductive tests. So we're kind of covering most body systems and disease areas in some aspect. This is just to say where the portal is. It's at massphenotype.org. So here we collect together all the raw data, the statistical analysis and the data integration, as well as information about the project. And at the bottom here, so in orange and blue, these are the number of phenotype genes and lines. So you can see over the last few years, the number of genes and lines has increased markedly. So in latest release, we're around 5,000 phenotype genes now. And this represents 59 million different raw data points. And there's approximately 358,000 different images available there. And from the statistical analysis, we identify almost 61,000 different phenotype associations to these lines. And these are encoded using the mammalian phenotype ontology. One thing I left off this slide is we also kind of capture those negative phenotypes, so we know what tests we've done for a particular gene and what wasn't significant, so that can be useful information as well. And there's actually a lot more negative phenotypes, as you'd imagine. I guess the biggest selling point of the IMPC is the new functional knowledge. So we've prioritized genes in the ignorome to date. So we've sort of characterized these as sort of genes where there was little or no previous functional knowledge. So we're trying to shed light on these so they may become new drug targets. This is kind of quite useful for the pharma industry. For instance, 55% of the genes, at least in release 5, had never had a mouse produced before. And then for 46% of the genes, there was never any experimental confirmed data in the gene ontology functional annotations basically meaning that no one's published anything on this gene with an experimentally sort of validated functional association. As an example of this, and to sort of show the type of data that's available on the portal, I'll just show this gene LMOD1. So you probably no one in the room has heard of this gene before, because if you go to PubMed and search for it, there's only 6 PubMed hits, and all we know about it is it's expressed in the brain, and it's part of this GTPase activating proteins for the R family, but we really don't know what it does until we sort of run it through the phenotyping pipeline. And this is like the high-level heat map overview of what we found from the IMPC pipeline. So in blue and the orange circles is where we found a significant hit. And you can see we found hits in like adipose, behavioral, cardiovascular, cellular, growth, hearing, homeostasis, immune, nervous, and skeletal phenotypes. So all over the body system, all over the sort of potential disease areas. And you can drill down into each of these to see the individual genotype-phenotype associations, as well as like looking at the raw data and the box plots of the actual raw data. So here you can see for the acoustic startle tests, you can see the female and male HOMs have got a significantly decreased acoustic startle response. And you can sort of see the results of the statistical pipeline. You can also sort of like drill down for this gene to look at the image data, such as the X-rays. So that's not projecting very well, but it sort of shows a nice scoliosis phenotype. And the lac-Z expression, so here we got the brain, and confirming on the right-hand side, this is the mutant data showing high expression in the brain. So over the last couple of years, we've kind of got to the stage where we've got several thousand sort of gene to phenotype associations. We've got a large corpus of data, and we can really start to explore this and perform interesting data analysis. So we've been publishing on this over the last couple of years, revealing sort of novel features of the mammalian genome landscape. So one of these is like the disease model study that was sort of my group led on, and I'll sort of talk about in the next section. We've had a couple of papers really highlighting novel disease gene candidates for metabolic disease and deafness, and some really interesting studies on sexual dimorphism and essential genes, which I'll give a few examples from. And this is just detailing these studies that have all been published literally in the last couple of years in high-impact journals such as Nature and Nature Genetics. And we've got sort of several more papers in the pipeline. So just to show you like the sort of key figure from the sexual dimorphism paper. So you can see at the top, this is looking, because we look at 7 male and 7 females, we can really start to look at this in a sort of large-scale way, all those parameters we measure. So this is looking at just the wild-type control mice, and we find in the categorical variables that we measure, like 10% of the data shows a difference between male and females. And this really kind of highlights like why in all biological research you probably want to be using male and female animals or even human patients. Yeah, a lot of drug studies, and for instance, are not particularly doing this and looking at male and females. And then the continuous variables have even greater resolution, so more than 50% of the data, the parameters we measure show a sort of sexual dimorphism. And then when we look at our phenotype calls that we call from our mutant lines, we sort of see the same thing. So for the categorical data, in 7% of the data, the male shows a greater effect, and 6.5% the female shows a greater effect. And then for continuous data, we can break this down even further, where we can start to see in 13% of the data, we only see that phenotype association in one of the sexes. And even if things like in 3.5% of the data, we see a completely different direction phenotype abnormality. So we'll see like an increase in one particular parameter in the male and a decrease in the females from exactly the same knockout. This slide summarizes what we found from our sort of developmental embryonic study. So this was based on the first 410 embryonic lethal lines that we produced. So a lot of these were novel genes, and we managed to assign novel gene function to these genes, particularly from the 3D imaging studies. And we're able to really start to quantify incomplete penetrance by looking at the subviable fraction. So we find 10% of our lines are subviable, so make it all the way to adult and we can phenotype them. Whereas like some other mice with exactly the same mutation in a single genetic background die. So we can start to study this incomplete penetrance. There was a high overlap with human essential genes, as you might expect. And the kind of interesting thing that we're studying further is this enrichment for disease genes in the mouse essential gene fraction. So here, this is the proportion of disease genes in the HGMD database. So if you look at all protein coding genes, it's about sort of 80%, as I showed for earlier. But if you look at the mouse essential genes in red versus non-essential genes in green, there's a big enrichment, statistically significant, for disease genes. And we're starting to drill down into this in more detail. Another aspect we're introducing is sort of late adult phenotyping. So we do the whole adult phenotyping pipeline and then carry on aging the mice up to a year old, and then repeat the whole adult phenotyping pipeline to try and discover differences that will correspond to late onset disease. And because we're doing this aging period, this gives us a much bigger window to introduce some new tests, such as home cage monitoring. I guess the biggest sort of game changer, like the last year or two, is starting to interact with some of these large-scale human disease sequencing studies, such as the 100,000 Genomes Project, and then the Precision Medicine Initiative in the US and the Centers for Mendelian Genomics. So there's a lot of obvious synergy between what we're doing and what they want to find out. So they can use our data to validate a de novo potentially in a completely novel gene that's not associated with disease that they might suspect is a new disease gene. We can help them to produce, using CRISPR-Cas9, exactly the same mutation in mouse and put it through our same phenotyping pipeline to compare with what we see for the null. And what we're finding more and more is we're starting to collaborate on big data studies. So we're producing this really big genotype to phenotype data set in mouse. Other people are performing disease sequencing and producing the same genotype to phenotype data sets in human, and such as the 100,000 Genomes Project, UK Biobank. And by sort of putting these data sets together, there's a lot of potential for machine learning. So this sort of kind of summarizes some of the interactions. So we're kind of seeing ourselves sitting in the middle here as a key role, playing a key role for mammalian functional genomics, because I think we're sort of seen as we've got these gold standard mutants. We've produced very reproducible data. We're kind of very strict about our protocols and the statistical analysis. So on the left, we interact with a number of the model organism communities, particularly via the MGI group at Jackson Laboratory, the Monarch Initiative and Marvel sites. And then on the right-hand side, we have all sorts of disease sequencing projects, such as run by Genomics England, or the Undiagnosed Disease Program, or Undiagnosed Disease Network. And then just to go back to that point about offering this capacity to phenotype precise human alleles. So if a human geneticist or someone like looking at the 100,000 Genomes Project, if they're interested in a particular loss of function mutation, they can see if there's a mouse line and data already available in the IMPC. But if there's not, they can nominate it for production, and it'll sort of rise further towards the sort of top of the priority list. Obviously, we're going to do all genes eventually, but if someone's particularly interested in the gene, we can prioritize it. If it's a missense mutation that they're interested in, there's a small chance there might be a relevant mass line already available. But generally, we'll want to produce the mass version of that point mutation and phenotype it. So various of the IMPC centers are setting up pipelines to do this, and in the UK, MRC Harwell have set up this genome editing mice for medicine call, where people can nominate to have some of these point mutations made in the mouse. And we're particularly using this for Genomics England to validate some of the 100,000 Genomes Project mutations. So moving on to some of the disease models from the IMPC. So like back to this slide, I sort of showed that PAX6 mutations result in I mutations, but there's subtly different mutations. So in the human, you've got an absent iris and opaque cornea and lens, whereas in the mouse and fish, the eyes decrease in size, and in Drosophila, it's completely missing. So we want to be able to capture these differences and similarities, and to do this, we use ontologies. Ontologies represent knowledge into a structure where we can computationally find that particular terms are in common and score how similar phenotype matches are. And one of the challenges is in the human side, we use the human phenotype ontology to capture rare disease phenotypes, whereas on the mouse side, we use the mammalian phenotype ontology. So with my colleagues in the Monarch Initiative, we've had to develop various methods to map between these ontologies. But in short, I won't go into the detail, we've developed methods where you can take a patient in the middle here, represented by these phenotypes, which are probably a bit hard to read, but this top one's microcephaly. And then we can query the whole of OMIM and OrphaNet to say, are there any similar diseases to do differential diagnosis? Or we can query all the mouse models in MGI or the IMPC to say, are there any similar mice? And then if this mouse involves this particular gene, that becomes a candidate gene for this patient's disease. And we use these algorithms within the IMPC, so we compare all possible diseases in OMIM and OrphaNet against all our IMPC genotype to phenotype associations and look for like similarities. So this shows one example for gray platelet syndrome, where the known gene is MBL2, and when we look at that gene in the IMPC, it gets a sort of nice phenotype similarity score. And you can start to drill down into the results on the IMPC page, and you can see it scores well because it's reproducing the thrombocytopenia phenotype and the spleen phenotypes, and you actually can click on any of these blue squares and really see why it matched phenotypically. So we published a paper last year in Nature Genetics where this is like the high-level overview of this phenotype matching between diseases in the IMPC lines across the first 3,328 genes that were disrupted. And what we showed in this study, so there's 889 Mendelian disease gene associations where we have IMPC data for that gene in the mouse. And we found for 41% of them, we have some sort of phenotype similarity between the mouse and the human disease. I should say, like, this was like incomplete data on the IMPC side in some cases, so we might expect this figure to go up in the future. But the majority of these 360 new models that we found were completely novel, so there'd never been a mouse published before that claimed to sort of model this particular human disease. And this is just a summary showing like we found models across all sort of major disease areas, body systems, from bone, cardiovascular through growth, body size phenotypes, hearing, blood, metabolism, muscle, and eye phenotypes. So just to highlight a few examples of this. So Bernard-Soulier syndrome, a set of autosomal recessive bleeding disorders, and they're all caused by mutations in the von Willebrand factor receptor. And this receptor is a protein complex composed of GP1BA, GP1BB, GP9, and GP5, so glycoprotein 9, GP9. And we've produced an IMPC mouse on this, sort of nicely sort of phenocopied the phenotypes you see in the human disease of prolonged bleeding time and large platelets and thrombocytopenia. So this is the IMPC data, and you can see in the mutant here on the right, increased platelet volume and decreased platelet numbers. There's a similar example, so Bardet-Biedl syndromes, which are a set of recessive ciliopathies caused by mutations, and here there's like any one of 19 genes that form this BBS-some. So one particular gene, BBS5, we found the mice recapitulate the retinal phenotypes and clearly the obesity phenotype shown over here on the right. But they kind of show some other interesting changes, such as impaired glucose homeostasis that's not described in patients, but maybe it's an interesting thing to look into the patients. And the final example, human phosphorine phosphatase deficiency in the PSPH gene. So if you sort of look at the literature on this, it'd be likely lethal in human without medical intervention. So this is one of the challenges comparing the human and mouse data. Obviously, we don't do a lot of medical intervention on the mouse lines. It was homozygous lethal in the mouse. And then when we look at the embryo data and the micro-CT images, we actually find very similar developmental and structural abnormalities to those seen in the adult human disease patients that obviously live because they have this extra medical intervention. Okay, so I'm going to sort of briefly cover the MONIC initiative. So the MONIC initiative is, I think I've sort of said before, is behind like a lot of the data ontologies and software that underlies all the data I've shown so far. And the role of the MONIC initiative is to sort of go beyond the human and mouse and sort of bring in data on as many possible species. So the more species you bring in, the more you cover those gaps in knowledge where you've not got data on the human gene. And I've just got like one overview slide on MONIC. So it's collecting data from a massive variety of sources. So there's sort of 23 different diverse sources of data based around genes, such as sequences, variants, and all the orthologs. There's lots of mechanistic and context data, such on pathways and functions, and 18 different types of phenotype and disease data, so as well as the phenotype annotations, the severity, and the temporal progression. And as I said, for all different species. And the way this works is everything's mapped to ontologies, so the Monarch Initiative also maintain a lot of ontologies, such as the Mondo disease ontology, this Uber-pheno cross-species phenotype ontology, for instance. And this allows you to sort of query across all these diverse data sources and to build some of the tools that I'll sort of talk about later. Yeah, and then perform queries like this. So this is just a screenshot from the Monarch site where you can look at one particular disease, such as neuronal ceroid lipofuscinosis, and see all the, so these are the human phenotypes associated with this disease down here. And then over here you get a sort of phenogrid map of all the human disease matches to those phenotypes, all the mouse phenotype matches, and then sort of fish, fly, and worm matches, because they're going across species. And then I want to sort of just finish off by talking about the role that sort of clinical model organism phenotypes play in rare disease diagnosis and gene discovery. So as part of the Monarch Initiative, what we're really trying to do is, so this very sort of challenging trying to diagnose disease patients just using the genome data, as I'll sort of show on the next slide, I think it is. So like bringing together the patient phenotypes and public reference databases of genotype to phenotype associations can be useful. And then moving forward, we really want to start bringing in other omics data sets, and possibly when we move on to common disease, the environmental factors, to like improve our ability to use genomics to diagnose patients. So the first version of this is the Exomiser software that my team and others in the Monarch Initiative have developed. So the idea behind Exomiser, so we start off with a whole exome, or indeed a whole genome, where these black bars are genes and the sort of stars represent variants. So for a whole genome, you're sort of talking about sort of 5 to 10 million variants that you're trying to filter through and find the single one or two variants that are causing the rare disease. You can do the standard stuff using the software, so you can remove all the non-coding, any common variants above a certain allele frequency in Nomad, for instance. And if you've got family members, you can do Mendelian pedigree filtering to sort of get that, filter out many variants. But what you find is you're still left with sort of tens or hundreds of variants. And even if you use the allele frequency and predicted pathogenicity from programs like CAD or Polyphen2, you seldom get down to a single striking candidate. You're generally trying to bring in some other information. So on the right-hand side, this is the other information we bring in Exomiser. So we can compare the patient's phenotypes to these reference databases of phenotypes from human disease, so like all the OMIM and OrthoNet phenotype associations to diseases and genes. And then the model organism phenotypes that I've been talking about today from the IMPC and then zebrafish resources. Even then, there might be some genes with no phenotypes, and we can use protein-protein interaction networks to fill in the gaps in this sort of kind of guilt by association type mechanism. And by combining all this data together, what we sort of hopefully end up with at the bottom here is a sort of single variant that's really rare, segregates with the family. It's predicted to be pathogenic, but it's also in the gene where the patient's phenotypes look similar to previous knowledge about that gene from model organisms or from disease databases. So when we sort of first published this, we kind of did benchmarking on simulated exomes where we put like known disease variants and phenotype data into 1,000 genomes, kind of unaffected exomes. And we can show the software in this like ideal scenario obviously performs very well. If you just look at the black bar, this is the percentage of cases where we get the right answer as the top hit. So you can see, most of the time we're getting towards 100% of the time we can get the right variant as the top hit in this ideal scenario. And even when sort of trying to discover novel associations, so where we kind of mask out the known disease gene association, we can do really well. If over sort of between 50 and 70% of the time we can get the right variant as the top hit. And this performance is sort of largely driven by this mouse genotype to phenotype data. So just to sort of show you a couple of examples of this this is a collaboration we had with the Undiagnosed Disease Program, where we were running Exomiser on their cases. So this was a previously solved case from the UDP, and these are the patient phenotypes: osteopenia, short stature, hyperglycemia, and acidosis. And we found a variant in this SMS gene was the top hit by Exomiser because, first of all, it's shown to be rare and pathogenic, but also the phenotypes were very similar to the patient phenotypes when the Exomiser automatically looked at the IMPC phenotypes. So, we have decreased bone mineral density matching osteopenia, decreased body length matching short stature, and the same sort of glucose phenotypes. And an example of like a novel discovery that we made with the UDP. So there was one patient there was two patients with variants in this STIM1 gene. So they were considering it as a candidate, and when we ran Exomiser, it was the top hit because there was already an existing mouse model that had been published in the literature in this paper here that showed very similar phenotype abnormalities to the patient. So there was the abnormal platelet activation and the thrombocytopenia phenotypes being picked up. So we now, as part of my sort of like other sort of day job, as part of the 100,000 Genomes project, we run Exomiser on the cases. So this is just showing the performance on 263 of the solved cases from the project. So these are like all the cases where we had not run Exomiser initially. And these represent a whole range of disease categories, from like some of the easier ones to solve, such as intellectual disability, to kind of way more challenging things like familial colon cancer. And what we've managed to show is in almost 70% of the time, the Exomiser top hit identifies the solved diagnosed variant. And it kind of rises to over 80% if we look at the top 5 hits. And a lot of the time when we're missing hits is because of incomplete penetrance, which is something to address in future versions. And then I think the slides will be shared anyway on the conference website. So this is just a sort of reference list for some of the Exomiser publications, but the key one is like how to use it, which was published in Nature Protocols. And the other ones are just the various iterations of the Exomiser software suite. So the first publication in genome research, the latest algorithm was in genetics and medicine. Also in 2016, we published a Genomiser, which takes the same concept but looks at non-coding variants as well. So we can start to look at variants in enhancer regions, for instance. And then Phoenix just looks at human disease, so it doesn't take into account the model organism phenotypes. And Exome Walker just looks at protein-protein interactions. So in summary, I kind of hope like I've managed to convey that mass phenotypes are available for many genes where we don't have data from the human, at least on single gene disruption studies. So you can use this mass phenotype data, and particularly the robust reproducible data that's stored in the IMPC, to sort of drive your functional experiments, so to come up with new things to test. And also, if you've got a variant in the gene that you know nothing about, it's well worth looking at these resources such as IMPC to see if it confirms the patient's phenotypes. And then I've sort of shown how IMPC is generating the first complete catalogue of mammalian gene function. So just like by the end of 2018, we'll hope we're going to be a third of the way there. And obviously all this data is produced in a standardized, reproducible, and most importantly, a non-hypothesis driven manner. So we do the same tests on all genes. And then the cross-species phenotype comparison tools that have arisen from collaborations with the Monarch Initiative allow us to detect these new disease models in IMPC. So, and then people can go ahead and order these mice for mechanistic and therapeutic studies. And then this software particularly allows us to automate variant prioritization, so in a diagnostic or discovery setting for large-scale disease sequencing programs, such as the 100,000 Genomes project. And then I just want to sort of advertise if you want to find out more about IMPC. So Pilar from my team is giving a talk on Monday afternoon, and she's going to go into way more detail on how we do the disease model pipeline, and particularly some compelling new disease models we found since publishing that paper in Nature Genetics. We've also got a booth in the exhibition centre, so we've got a nice new booth to show off at booth number 280, so please come and visit us. I think we're going to be there the whole time. And then Violetta's got a poster That goes in, again, sort of summarizes the whole of the IMPC. So if you miss us at the booth, come and talk to Violetta, or the rest of us will probably be standing around it at the time as well. And then just to sort of finish off, there's obviously like way too many people involved in the IMPC to name them all. So just to sort of thank the funders, like the major funders, particularly such as the NIH and the European Commission, and all the different centres involved in the mass production, phenotyping and data analysis. And then from the Monarch, again, there's too many people involved. So these are just all the key sort of partners and stakeholders and people who contribute data. And with that, I think I'm done, and I'm sort of happy to take questions.
Thank you very much.